Ma'am, can I start?
Yes, please.
Welcome back once again. Now, I introduce you all to today's speaker, Dr. Pamita Avasthi. She is working as an associate professor at NIT Hamirpur, Himachal Pradesh. She did her BSc honours from St. Biddy's College, Shimla in 1992 and completed her MSc and pre-PhD and PhD from Himachal Pradesh University, Shimla in 1994 and 2000 respectively. She did her postdoctoral research from Department of Biotechnology, IIT Roorkee from 2000 to 2003. Dr. Avasthi has been working in Department of Chemistry, National Institute of Technology, Hamirpur since 2005. Presently, she is working as associate professor in the same department since 2012, teaching undergraduates BTech and postgraduate MSc students. She has completed 11 PhDs, 17 master projects, and 3 undergraduate projects. 2 postdoctoral fellows, 3 PhDs, and 3 master's dissertations are going on. Dr. Avasthi has handled 7 R&D projects as a postdoctoral fellow, as a PI, and as a scientist mentor. She is also invited in external funded entrepreneurship and innovation projects as principal investigator, NITH, ITBI, Department of Science and Technology, Government of India, and nodal officer, CM Startup Project, Government of Himachal Pradesh. Dr. Avasthi has organized 27 short-term courses, workshop, conferences, and attended 26 workshops and training programs along with 15 expert lectures has been delivered till date. Her research interests and areas include natural product mimics, bioorganic and structural chemistry, synthesis and biological evaluation of analogues of mitozintron as a potential anti-cancer agents, interaction of mitozintron and its synthesized analogues with DNA sequences using 2D NMR, UV visible spectroscopy, fluorescence spectroscopy, cyclic voltammetry, viscosity and molecular modeling tools in order to design and develop new class of anti-cancer drugs. Synthesis, structural study using IDNMR, FTIR spectroscopy tools and mass spectroscopy, etc. Biological study and field evaluation of sesquiterpenoid, a class of juvenile hormone analogues containing diverse structural features as a potential environment-friendly green pesticides, natural product mimics, and quadruplex binders. Therefore, I request ma'am to kindly give us your respective talk.
Thank you, Simran. Once again, a very warm welcome to present here in online mode. And I would request Ravinder to check whether the online streaming is there because some message had come that online streaming has been stopped. So please check that and also the recording of the lecture. So, as thank you, Simran, for your uh, humble introduction about me. I'm a very small person. I'm trying to find out my mission in life. Anyhow, thank you very much for this. HICT, please switch off your speaker. Uh, I I welcome Professor Berthwal, my professor, my mentor, my guide. She's also online. Thank you very much, ma'am. I'll try my level best to introduce or about what are these DNA quadruplex structures and how do we read it. Though there is a lot and lot of information available online or in these platforms, these virtual platforms, but I'll just try my level best to give you the concrete and what they are and how do we study them, how do we read them, because we all know what DNA is and what are their various applications, etc., etc. So let us start with it. My presentation is moving. Second slide is there.
Yes, ma'am. Second slide is visible.
Okay. If there is any gap, please let me know. Right. So, as a chemist viewpoint on the function of molecule or a system of molecule, the question is how molecular structure dictates the function as a chemist point of view. The most DNA structure widely organized as a classical DNA double helix, which defines a structural basis for the genetic code. DNA is structurally dynamic and capable of adopting a different secondary structure. One such class of DNA secondary structure is the four-stranded GNA quadruplexes, which is also called as G4 tetrad as a one of the unit. Herein, in this talk or in this lecture, we are going to discuss some of the key scientific history that has shaped our understanding of the structure of G4 DNA and its dynamic. The study of G quadruplexes that began in 1910 with the observation. Slide is moving.
Yes, ma'am. Slide is moving.
Okay. In 1910, with the observation that high concentration of guanylate acid, which is also called as guanosine monophosphate GMP, this is the molecule which formed a gel. GMP is composed of guanine, which is a nitrogenous base, ribose sugar, and a phosphate group. Components of RNA, as a component of RNA, it involves in various cellular functions like protein synthesis and gene expression. It is also a key player in energy metabolism and signal transduction. Industry, it is being used as a flavor enhancer. And industrial production is generally done by RNA breakdown, nucleotide extraction, or by microbial fragmentation, etc. It is in 1910, the Bang reported that guanylate acid solution at high concentration formed a gel, making the initial observation of G4-related self-assembly. In 1962, the G-tetrad structure, the building block of G4, was determined through X-ray fiber diffraction studies. G4 once viewed primarily as a structural feature of DNA and RNA. However, research has since revealed their involvement in various biological structures, and a lot of work is reported in this particular area. G4 are also known to play a vital role in transcription, replication, genome stability, and epigenetic regulation. G4, this is our area of research. G4 has been linked to a cancer, particularly the telomeric length, maintenance, and telomeric activity. So what it is basically, the four guanine bases form a square planar array where each base is both hydrogen bonding, acting as a hydrogen bond donor as well as hydrogen bonder acceptor. Engagement of both nitrogen, that is N1 and N2, of one phase with the O6 and O7 of the second phase on guanosine yields a total 8 hydrogen bond as per planar quadrat. So this is called as one quadrat. With the development of chemical synthesis of extended polyguanine oligonucleotide strands, observed additional structural information, and same is further confirmed by various spectrophotometric techniques like CD, IR, NMR, and X-ray crystallography. X-ray crystallography analysis particularly indicates that four strands motif, which is stacked upon one another, and whole of this molecule is called as a quadruplex. And how these techniques they are being used to deduce this structure, we would be having in the subsequent lectures in subsequent days. So these stacked tetrads, they align themselves to give similar appearance like that of DNA. So research in the area of G quadruplexes was ignited early in 1990 by identification of the G-rich repetitive sequences located at the end of the chromosome by Blackburn et al. In Jogal's research group. These guanine-rich repetitive uh, structures in telomeric DNA sequences could form a high-order structure and likely to involve in chromosome maintenance. So before we go into detail, let us start with the chromatin itself. So DNA is not an isolated macromolecule within the nucleus of the cell. It is associated with variety of proteins such as the histones, and whole of this structure along with the protein is called as a chromatin. The histones and associated DNA form a structure is called as a nucleosome, which occurs regularly throughout the length of this chromosome and plays a very crucial role in DNA regulation, transcription, translation, etc. So the nucleosome and these are the telomeric ends. These are the important areas of structural studies. So let us have a brief discussion on the deoxyribose nucleic acid structure and function. Watson and Crick, you all know, solved the secondary structure of the DNA by building a model that fitted all the known experimental results. The structure consists of two chains arranged in double helix form. Double helical form. In the primary structure, DNA building blocks, they are linked together via nucleosides. These nucleosides, for example, deoxyadenosine, deoxyguanosine, deoxythymidine, and deoxycytidines. Each nucleotide is constructed from of two components. One is the deoxyribose sugar, another one is the base. Sugar is same in all the molecules. The four bases, two are bicyclic, which are adenine and guanine, and two smaller are monocyclic, that is cytosine and thymidine, and whole heterocyclic chemistry revolve around these molecules. So nucleoside building blocks, they are joined together through phosphate group which link 5-hydroxy. So this is the 5-hydroxy position and this is the 3-hydroxy position of the sugar ring. So these link through phosphate bonding. This is the phosphate group. 5-hydroxy of one nucleotide to the 3rd hydroxy of the next one. With only 4 types of building blocks available, the primary structure of DNA is far less varied than in comparison to the primary structure of the protein. So as a result, it was initially thought that DNA might be having a minor role to play in the cell biochemistry, as it was really hard to see that how such apparently simple molecule could have anything to do with the mystery of the genetic code. But the mystery of this genetic code lies in the secondary structure of the DNA, which has been proposed by the Watson and Crick. So Watson and Crick structure, in which these two DNA chains, as he proposed by crystallographic studies, that these two chains, they are arranged together in a double helix format with the constant diameter and having a major and the minor groove. Which these major and minor groove, they provides a platform for number of proteins and enzyme for various biochemical processes going in a cell. So this structure is relies crucially on the pairing of the nucleic acids between the two chains. Adenine pair with the thymine and cytosine pair with the guanine. In cytosine guanine pairing, we have a 3 double bonds. In case of adenine thymine, we have a two double bonds. Bicyclic always linked to the smaller monocyclic nucleotides. So this double helix, this is being stabilized by the fact that base pairs, they are stacked upon one another, allowing the hydrophobic interaction between the faces of these heterocyclic, because this is the highly electron rich area. The polar phosphate backbone is placed towards the outside of the structure, and it can form a favorable bonding interaction or polar interaction with the water molecule. Further, regarding the passing of information, how it happens, it is now possible to see that how the replication, that is the genetic information, is possible. If the double helix untangle itself, a new chain is constructed on the original chain, or in other words, original chain act as a template for the construction of new and identical double helix. So the template is being exposed to the bases, a template with exposed bases, this one, can pair by hydrogen bonding with the individual nucleotide in the form of a triphosphates. Once the nucleotide is base pair, I'll go ahead. Once this bond formation is there, so here the new nucleotide is coming. Once this is the exposed or the template chain. Once this bonding interaction takes place, the enzyme catalyzed reaction will take place, where the new nucleotide, this one, this is the growing chain, the new nucleotide is linked to the growing complementary chain with the loss of diphosphate group, which is acting as a good leaving group. It is important to note that the process involves each nucleotide reacting with the 3' end of the growing chain. And via this process, so this is the way the chain is growing, and details can be very well explained in cell cycle analysis, that is in a biological concept. So via this process, the genetic information is passed from one generation to another. So another question arises, that how can only 4 nucleotide code for a 20 amino acid? The answer lies in the triplet code. In other words, amino acid is coded not by one nucleotide, but it is set of 3, and there are 64 ways in which 4 nucleotides they can arrange in the set of 3. But in reality, DNA extremely is an extremely long molecule. It is so long. This is the structure of the cell, and here we have a nucleus. It is so long that it would not fit into the nucleus of the cell if it exists as a linear molecule. So it has to be coiled, right, into a more compact three-dimensional shape which can fit into the nucleus, and whole of this process is called as a supercoiling. DNA has to do a number of functions, replication, transcription, etc. So this process regarding the uncoiling or supercoiling, uncoiling, coiling, supercoiling, this process require action. Though there is a group of or a family of enzymes are there, but the main process require the action of the family of enzyme, which is called as a topoisomerase. Here I have notioned it as a topo 2 or topo 1, which can catalyze the seemingly impossible act of passing one stretch of DNA helix across the another stretch. So they do this by temporarily cleaving one or both together. strands of the DNA helix and create a temporary gap. Then resealing the strand once the crossover has taken place. So supercoiling allows the efficient storage of a DNA, but the DNA has to uncoil again if replication and transcription has to take place. If uncoiling did not take place, then unwinding process, which is catalyzed by the helicase enzyme that takes place during the replication and transcription, would lead to increase the tension because of increased supercoiling of remaining DNA helix. So it is a kind of a very coordinated process. So topoisomerase is a mammalian enzyme, so which is very crucial for uncoiling of the DNA. Enzyme binds to one of the DNA double helix, and it is the temporary covalent bond formation between the tyrosine residues of the enzyme with the 5' end of each strand and then stabilizing it. Strands they are pulled out, DNA does its function, and again it detaches itself from this position, and again the DNA takes up the compact or again supercoils and go back to its position. So this is the complete process, and whole of this structure is called as the ternary structure of the DNA. So early X-ray diffraction studies of DNA revealed that there are two kinds of structures, the A form of DNA and B form of DNA. The B form of DNA, this is the B form of a DNA, has 10 bases per turn and a wide major and narrow minor group, whereas A form is observed under the condition of low humidity having 11 base pair per turn. Its major groove is narrower and much deeper than that of the B form. Its minor groove is broader as well as shallower. The vast majority of the DNA in cell is in the B form, but it can adopt the A structure in certain DNA protein complexes. Also, A form of the structure sometimes RNA also adopts when it goes into double helical form. The DNA containing the alternating purine pyrimidine residues, which can fold into right-handed as well as the left-handed helix. In order to understand the folding, we need to consider that how this particular bond, the bond between the bases as well as the sugar molecule, position itself. This bond can have a two conformation, syn as well as anti. In right-handed DNA, we will discuss in detail in coming slides. The glycoside bond is always in anti-conformation, and in left-handed helical structure, the fundamental repeating. So, left-handed structure, specifically in Z type of DNA, these fundamental repeating unit usually is purine and pyrimidine dinucleoside, where this glycosidic bond is in anti-conformation at pyrimidine residue, whereas in syn conformation in purine residues. So it is this syn conformation at the purine nucleotides that is responsible for the left-handedness of the helix. So change to the syn position in purine residue to alternating anti-syn conformation gives a backbone of the left-handed DNA, a zigzag look. So the Z form of DNA, which can be distinguishable from the right-handed DNA forms. So now I'll go more deep into it. So there are two phases when the bases they are involved in hydrogen bonding when there is a hydrogen bonding between the two strands, right? So hydrogen bonding of the nitrogen bases, this is the Watson and Crick phase and this is the Hochstein phase. So as we know, the cytosine forms the three hydrogen Watson and Crick hydrogen bond with the guanine, and adenine forms two hydrogen bonds with the thymine. Tetrads, because we are talking of a double DNA strands, the tetrads, the G tetrads involves these two bases N2 as well as head. So this is an additional hydrogen bonding which is really critical for the another type of structure, the secondary structure, that is quadruplexes. And yes, all these hydrogen bonding together stabilizes the tetrad. So with the change in the pH, bases with the nitrogen atom have the added capacity to change the protonation state. At neutral pH, the standard base pairing would be there, but when the pH is greater than 9.2 or less than 4.2, the additional association will take place. Definitely changes in all these conformational angle, whether it is bases, sugar, or backbone, that will take place. So dihedral angle, what I'm talking of, the glycosidic angle, that is chi. This is the chi which is joining the sugar molecule with the nitrogenous bases. This is being defined as, this is the O4 prime, O4, C1, N9, and C4 or C8. So this is the dihedral angle, this specifically this one. This is the dihedral angle for purine, that is for bicyclic bases, and for pyrimidine, it is O4, C1, N1, and C2 are important in determining that how the DNA fold along with the stability of the DNA structure. The two most common conformation observed in the folded DNA structure, they are the syn conformation and the anti-conformation. Then comes the pentose sugar, that is the ribose sugar, which is also play a very, very important role in defining the secondary structure of the DNA. So these are non-polar, because you could see any double bond over here. So these are non-polar with number of energetically stable conformation. C2 prime endo. So look at the numbering. This is oxygen. This is C1, C2, C3, C4, and C5. Oxygen, C1, C2, C3, and so. Sorry, this is C4, C4, O4, C1, C2, C3, and C4. So C2 prime endo, how do we define C2 and C3 prime endo? So this is the orientation of these carbon molecule with respect to this one, and according to this carbon atom as well as the oxygen. If this C2 is above, then it is endo, C2 prime endo. If this is below, then this is, if C3 is above, then it is called as a C3 prime endo, or we can say C2 prime endo or C2 prime exo, or we can say C3 prime endo, exo, or C3 prime endo. So anything according to our this thing we can confirmation we can write. So C2 prime endo and C3 prime endo confirmation are the most important confirmation observed in DNA as well as RNA, and specifically in RNA, because this is for the RNA, this is for the deoxyribose. Deoxyribose means you don't have a OH group here. In ribose, you have a OH group here. So in case of RNA, sugar conformation is strictly restricted to C3 prime endo. Reason being because of the steric hindrance caused by this OH molecule present on this pentose sugar. So removal of this hydroxyl group from here and converted into deoxyribose, the additional energy barrier means more energetically favor structure is being formed. That's why DNA is having so n number of conformation possible. The energy barrier between C2 prime and C2 prime endo sugar pucker is approximately 8 to 1 kilojoule. So bDNA, if we talk of a bDNA is basically a C2 prime endo conformation, and C2 prime endo conformation is also observed in where is that? In G-tetrod also. So what I'm talking of? I'm talking of the the sugar conformation in bDNA as well as in quadruplexes. This is same. So besides strong linking between the sugar molecule and the nitrogenous bases, the this chi angle, the chi angle, is further extended to C3 prime and C4 prime atom of the sugar molecule, which is further connected to the phosphate backbone, or which is a part of the backbone structure. Sugar does not have any protonation site. However, O4 can extensively be utilized for the hydrogen bonding with the water molecule, which add extra stability to the structure of the DNA. So DNA backbone is having 6 atoms and backbone conformation. So we have a 6 atoms starting from here. This is 1, 2, 3, 4, 5, and 6. So all these dihedral angle, these 6 atoms, these atoms, they are being defined, or conformation around this segment is defined by the 6 dihedral angle, that is alpha, beta, gamma, delta, eta, and epsilon. So it is the alpha, beta, gamma, and this angle, they are restricted to energetically favorable orientation, that is minus 60, 60 to 180 degrees of angle, and it would be still expected that large number of backbone conformation would be observed in the nucleic acid because of this flexibility. And yes, stabilized structure, secondary structure, is the combination of the conformation of the backbone, sugar, as well as the nitrogen bases and its bond formation with the another strand, hydrogen bonding with the another strand. So due to this structural flexibility, I will conclude in this slide. So due to this structural flexibility, which is important for the DNA to function, so there is a great deal of structural diversities available to the DNA. One of the unusual polymorph is what we have discussed over here is the zDNA. This form has a left-handed helical rise and alternating, as discussed, the syn and anti-glycosidic angles. Transition from beta to Z form DNA has been studied by the X-ray crystallographic tools. Interestingly, in addition to, so this is the bDNA. bDNA can be transformed into the aDNA and as well as the zDNA, and all these structural conformation has been studied by, or has been confirmed by, the X-ray crystallographic studies as well as the NMR studies. So in addition to single strand for the DNA as well as for the RNA, double-stranded DNA, which has been very extensively studied, there is a 3-strand DNA also, which is called as a triplet-stranded DNA. So association has been termed as a triplex. There is an inclusion of the 3rd strand. This is the 3rd strand. It is here. This is the 3rd strand into the major group of duplex DNA by utilizing the additional hydrogen bonding and protonation states of the bases, right? And this is the quadruplex, and quadruplex is the most diverse polymorph of the DNA with 4 strands of DNA. However, the characteristics of the quadruplexes, they are similar to their double-stranded DNA form. So here in this table, I will just try to compare. So let us see this one only. What is the helix type? If you see the bDNA, it is right-handed, and this is the quadruplexes. Quadruplexes can be parallel, can be anti-parallel. So what is anti-parallel and parallel, we would be discussing subsequently. So you can see the helix orientation is right-handed in quadruplexes also. Number of bases per turn, you could see 10.5 in BDNA, and here also it is somewhere around 12 and 12, and also in anti-parallel conformation, it's also 12. C1 prime to C1 prime means C1 prime of one sugar from one strand to another strand distance is somewhere around 10, and it is 16. Most important iron overlapping factor with the BDNA is the sugar puckering, C2 prime in case of BDNA and C2 prime in case of the quadruplexes. And over here also it is C2. So two overlapping factors, that is the helix as well as the sugar puckering, more or less same in case of BDNA and quadruplex structures. So finally, so like BDNA, if we try, because if in this particular slide we would be discussing the comparative wheel plots distribution of various dihedral angles, what we have discussed over here, the chi starting from alpha, beta, gamma, delta, as well as the sugar buckling, etc. If we need to compare these angles, so wheel plot has been drawn. They are there to compare uh, various dihedral angle. Uh, for BDNA from two tetrad G-quadruplexes DNA conformation. This is, I think, anti-parallel. Yes, this is anti-parallel, and this one is the parallel. Parallel means how the backbone has oriented itself with respect to rest. So these are the parallel backbones. This is the one or two. They are anti-parallel. So these are the crystal structures. These are the results from the crystal structures of a quadruplex DNA, which has been solved for Oxygen DNA, and this is for human telomerase quadruplex DNA. So this is the PDB number I have given. So detail can be seen over here from the literature. So various dihedral angles, they are shown for each nucleotide. So you can say this is chi. You start from alpha, beta, gamma, delta, and so on. This is the 90 to 180, 180 to 270, then this is to 360, right? So so from these chart, if you compare BDNA with these and these two, in case of parallel, because you do have a polymorph structure, different conformation in the G quadruplexes also, so you can see there is kind of, compare only one for all the nucleotides. So single line is for one nucleotide. So you can see the if you just try to visualize the chi, you could see there is a overlapping between the duplex as well as the quadruplex in parallel conformation. But you could observe there is a diversity over here. So this is not overlapping. So just by change in this angle, the chi angle changes the conformation of the DNA in a different, or take it to a different level. So this can be confirmed from these experimental studies. So these are X-ray crystallographic studies on the various DNA structures of the human also, and other species also, whether it's a plant or animal. So, and what we could conclude out of it is that it is the flexibility on these dihedral angles which is the key factor in how the quadruplexes they are being formed, and how they held together, and how they fold, and how the supercoiling that takes place in the DNA. So this is the basics, that how the duplex differ from the quadruplexes, and how the quadruplex they are being solved on different DNA types using various techniques. The next part is, what is the stabilizing factor in the quadruplexes, because yes, they are the stable structure, and they are reported experimentally also. So what are the various stabilizing factor? Like double helix bDNA, bases in quadruplexes, they are associated by base stacking, hydrogen bonding, hydrogen structure, as well as electrostatic interaction. So one point to note over here is that it is in case of double helix DNA, we have a four bases. Here we have only one basis, that is guanine. So guanine is interacting with each other, forming this tetrad, and this tetrad is stacked one upon each other, and this is the orientation of the backbone. So till date, whatever the structures which are being reported in the literature, so orientation of this, stacking of these tetrads, and their connection by these backbones, they have been studied extensively. So let us compare with means how do we study in comparison to the stability what we have studied in bDNA. So bases in DNA, they show, when they are stacked, they show very unfavorable interaction with the polar solvent. They are associated and stacked upon one another to reduce or minimize the area they expose to the solvent. So that's why we say the core of the DNA is hydrophobic. Stability of this double helical DNA is being provided by the stacking of bases through the combination of hydrophobic, electrostatic, and van der Waal forces. Due to, because they have electrocraft, we do have a heteroatom. As we discussed, these are the purine and pyrimidine bases, bicyclic and monocyclic bases, and entire heterocyclic chemistry, that surrounds on these 3-4 bases. So they do have certain charge. Because of this slight negative charge on these bases, they have a tendency to repel each other slightly. So that's why we have this type of orientation. If you see over, this is for the tetrad, but this is for the double helix DNA. So base stacking energy, which has been calculated, so I'm not going to that detail, is comparable between CG base pair as well as in AT base pair, and based upon that, whole of the structure is being stabilized. But as you see, these base pairs cannot sit directly on each other due to steric interaction, because very intense electron cloud is there, but they are twisted around the helical axis. So this is the helical axis, means the axis which passes the center of the molecule. Distance between the each pair is defined as a rise, while rotation about this axis is called as a twist. So further, other parameters such as roll, rise, right, tilt, roll, rise, tilt, slide, shift, they provide the additional description of one base with respect to other, and finally define this global helical axis of the bDNA. Change in roll, this as well as tilt, are responsible for the change in conformation from bDNA to aDNA. Further, bending and buckling effect is required, or you can say it is important for DNA to pack around histones and other proteins, and which ultimately gives rise to the tertiary structure of the DNA. Now, if you compare it with the quadruplexes, this is one guanine, or you can say this is guanine, this is cytosine, right? But over here, we have both this guanine. We have 4 bases. That's why it is called tetrad, right? Tetrad stacked upon one another. It is called quadruplex, right? So the stacked guanine in quadruplexes neither exactly symmetric nor or planar. Geometry of the G quadruplexes defined as per lines of the geometry which defines the double helical DNA, like slide, shift, roll, tilt, twist, and rise. Additional variation in tetrad, because we say we have a parallel, we have a anti-parallel conformation. Anti-parallel means the orientation of the backbone. So additional variation in tetrad geometry that comes from the stacking of, this is one guanine molecule basis. So this is one phase, one plane, and this is another just below it, right? So you have oxygen in red, it is being shown. This is a central coordinating ion. So geometry of the stacking guanine bases and alternating, that is syn anti-syn glycoside arrangement. This type of arrangement in quadruplexes is found in antiparallel. So if you have a alternating, syn chi, I'm talking of chi, we have discussed chi in detail. So if the chi geometry is alternating, that is syn, anti, or syn, then we have an antiparallel conformation. But if it is either syn or anti, then we have a this type of kind of a anti-parallel or parallel backbone arrangement. So in parallel standard structure, only, as I said, single stack arrangement with anti-glycosidic torsional angle has been observed. Variation in the basis. Now, whether it is syn, anti, or whether it is in parallel or in anti-parallel, what it is going to affect? It is going to affect the position of oxygen atom, right, which is opening towards the center. So you can see very well over here also, and also define the size of the cavity and the coordinating cation here. So what I can say in conclusion, that position of O6, this is O6. What is O6? I'll come in detail. This is O6. This is guanine. I'm talking of this O6. O6 on this nitrogenous base. So I'll come here again to this slide. So the variation in the configuration of the bases affects the position of the O6, which is further responsible for the formation of the cavity located at the center of the quadruplex cavity, and this might play a vital role in the metal ion selectivity. And this is one of the area which is specifically the latest research area in organic electronics. This area. This is another one of the application we would be discussing in subsequent lectures in the coming days. So next stabilizing factor is the hydrogen bonding. So hydrogen bonding is major mode of interaction in DNA. It controls both secondary as well as the tertiary structure of the DNA. In quadruplexes, in addition to Watson and Crick base pairing, what we have discussed, the Hookean base in G quadrate increases the potential number of hydrogen bonding to 8. Hence, the hydrogen bond energy association with the quadruplex formation increasing from 0.22 to 0.42 electron volt when involved in the quadruplex formation. Next is obviously the DNA is in chromatin, and chromatin, yes, so many enzymes, proteins, and you do have water as a medium. So hydration energy, or you can say, this is the another factor which stabilizes the quadruplex structure. So like duplex DNA, quadruplex have grooves. It has 1, 2, 3, 4. So they do have a groups and network of water molecule that is being formed there. These water molecules show very extensive hydrogen bonding with N2 exocyclic as well as N3 heterocyclic atom of the base. This is a fourfold symmetry which has been observed. In parallel hydration structure. So as I told you, we have a two type of orientation of a backbone in a quadruplexes. One is a parallel, another one is an anti-parallel. In a parallel, the hydrogen structure has seen, this is the structure by X-ray crystallography. The hydrogen structure, the four group has very well occupied, which stacked upon one another. However, in case of anti-parallel quadruplexes, with the alternating glycosidic torsional angle, that is syn, anti, etc., we see a more complicated and less order hydrogen structure. Sugar oxygen atom, I'm talking of this sugar oxygen atom, or this specifically this sugar oxygen atom, also associate itself as with the as a hydrogen acceptor and form a second tier of ordered water molecule in the grooves. Network of ordered water molecule highlighting the interaction of water with the N2, N3, as well as the O4 base sugar atoms to external phosphate oxygen, specifically in uh, X-ray or NMR salt structure of human telomeric crystal structure. So this structure has been taken from there. So network of water has been observed, which further stabilizes the structure of G quadruplexes. Another is the electrostatic interaction. Electrostatic interaction by the metal ion coordination. So O6 atom, these are the O6 atoms. This is the O6 atom. You can see forming a hydrogen, four hydrogen bond. This is the one tetrad, another tetrad, and then so on. And this is parallel, anti-parallel, and hybrid. We will discuss in the next slides. So oxygen atom forms a square pillar kind of arrangement for each tetrad with a twist of 30 degree and rise of 3.4 angstrom. So between each tetrad with bipyramidal antiprismatic kind of a structural arrangement where you can have a eight oxygen O6 atoms, they are being involved. So centrally located charged cavity are localized by the coordination of a cation. Selection of cation that is based upon the size and charge of the cavity, which ultimately responsible for the folding of G quadruplexes. Potassium ions effectively coordinate. This is the potassium effectively coordinate. You could see you have a four oxygen atom here and four oxygen atom here. So these carbonyl oxygen atom position at 2.7 angstrom from each oxygen atom. Sodium has also been observed in the crystal structure, which is slightly above and below the central position, which are very close to the tetrads or tetrad structure, G tetrad structure. Then comes the G quadruplex polymorphism, that is topology and structure. The how do they pack themselves, that how the packing of the secondary, tertiary, and tertiary into the quaternary takes place. So they are also broadly classified on the basis of combination of number of stacked G tetrads, polarity of the strands, location and length of the loop. So we will start first of all with the number of stacked G tetrads, which can be unimolecular, bimolecular, tetramolecular. When I'm talking of a unimolecular, I'm talking of a single strand. I'm in a bimolecular, two strand. Tetramolecular means four strands forming a G quadruplexes. And this is, you can say, um, this is the nomenclature generally being used. Specifically in unimolecular, if you see the unimolecular, in case of unimolecular, in this case, what does GM means? What does Xn mean? And what does this X naught mean? So M is the number of GM means, M in G means the number of G residues in each shot when it is folding trapped involved in G tetrad interaction. And Xn x naught and xp, that can be any combination of the residue, any combination of the residue, including G. So it can be A, T, or any residue, right? In case of bimolecular, which is also called as a dimeric, as well as a tetramolecular, the two strands can be equal or they can be non-equal sequences which has been observed till date by various experimental studies. Again, the same terminology where xn, what is the meaning of xn and x0 is same. It can be 0 in case of bimolecular or maybe 0 or may not be 0. हो सकते हैं और नहीं भी हो सकते हैं. Then comes the tetramolecular. In case of tetramolecular, there are four strands with this terminology. They arrange themselves. So broadly, the G quadruplexes, they are being categorized into unimolecular, bimolecular, and tetramolecular. Then comes the polarity. So the quadruplexes, they are further classified on the basis of the strand polarity, means I'm talking of the backbones, right? And location of the loops. Loops means which is connecting these two strands. So G, in case of when the G quadruplexes, they are either formed from a single strand or it is formed from the double strand. In case of a unimolecular, the parallel, this is the unimolecular. You can have a parallel, you can have a anti-parallel, right? So in case of parallel G strand requires a loop to connect, means when they are being formed, so it requires a loop, this is called a loop, to connect to the bottom of G tetrad. Hence, specifically this one, in case of parallel, this is called as a propeller type of loop. It is also called as a double chain reversal loop. So this is one tetrad. I will go more deep into it. Just try to visualize the two stacked G tetrad sequences. This is one G tetrad. This is another G tetrad. So the sequence is going something like this way. This is called as a propeller loop. So when it is, there are two steps. When you have a multi n is or m, m is more than 3, 4, 5, then the strand starting from here going in this way and this way and so on. This is called as a propeller kind of a loop, or also called as a standard reversal loop. So this type of structure feature has been studied both in X-ray as well as in NMR salt structures for human telomeric DNA sequence. So if one out of four, right, if one out of four is anti-parallel to the rest, so this type of topology is fine majority for biomolecular, right, as well as also found in the unimolecular quadruplexes, right? And in addition to the propeller, we do have the loops of lateral, which is also called as the edgewise. Lateral means this is the lateral. Somewhere it is called chair, or sometimes it is called as the edgewise. And diagonal loop. Diagonal loop means when this strand is connected to this strand, this is called as a diagonal loop. Or over here you can see this is the diagonal loop. Joins, which joins the opposite strand, either on same phase or in opposite phase, right? Or head to head or head to tail. So in more elaborate way, I will discuss it over here. So if you see, these are the various types of, you can say, the polarity at the backbone as well as the loop formation. You can see all these backbone strain. Green is syn conformation of the guanine bases. Yellow is the anti-conformation of the guanine bases. Direction is O5 prime to 3 prime. This is the backbone, and dot is the counterline. If you try to visualize these two structure, all are parallel, all are parallel. The G bases can be anti, it can be syn, right? Now, if you see these change in polarity over here is, here you have a two alternate, you can say the side chains. They are in opposite direction, so it is anti-parallel. Here one is in opposite direction, anti-parallel. Here in one phase, two are in opposite directions. In two different phase, this is in one phase. So if you try to in this direction, right, as well as in this direction it is in one phase. So when this happens, you can see the conformation of the glycosidic angle, that is a chi angle, that also changes. So here you have a alternate sin, anti-sin, and so on. You can have all anti, one sin, or two sin, or two, and this is the way the conformation or a polymorphism in DNA quadruplexes that can be observed. So this is sin and anti conformation. We have discussed all these in detail in the previous slides. So further, the loop formation means they are not in air. Obviously, they are being connected by X, Xn, right, etc., X0, XP. So these are the flanking nucleotides which are connected. This stand with this one. So this is edgewise. This is also edgewise, or two different faces. This is the external, lateral, diagonal, and external loop, which has been studied, observed, identified, or by various advanced structural tools, that is NMR and X-ray crystallography. So we will be discussing in brief I will take one example of 4-repeat human telomeric sequence. This is the structure which has been solved by NMR as well as by X-ray, and you can see the difference in the solution and by both the experimental techniques, right? In case of if you see here in this case, specifically this one, showing the intramolecular G-quadruplex geometry in sodium ion solution, in which guanine around each tetrad are sin-sin, this is sin-sin, and anti-anti. Red is sin, blue is anti. So this is the glycosidic means chi angle orientation, or you can say the conformation. And loops, they are successively. This is the edgewise. These are the two edgewise. This is the diagonal. So this is edgewise, diagonal, and again the edgewise. So this is the orientation of the you can say the G quadruplex or structural conformation of the G quadruplexes by NMR in the solution state. So each G tract, each 1, 2, and 3 has both parallel as well as the anti-parallel adjacent S. So this is parallel direction. You could see the direction of the phosphate backbone. These are two are parallel, and this one is anti-parallel. But if you try to observe the structure of the human telomeric sequence by X-ray crystallography, the same sequence, the presence of the potassium ion revealed a very different kind of intramolecular G quadruplex. All the intramolecular, in this case, all the guanine, these all guanines, they are anti. They have an anti-chi conformation. Loops, they are double chain reverse. That is a propeller kind of a shape. You can say this is a propeller kind of a orientation of the loops. And these structural features facilitates the high order telomeric folding and unfolding in chromosome. So what we had discussed in the first class at the telomeric junction. So these structures, they have been experimentally confirmed. So finally, methods of quadruplex structure determination, they are further classified. The computational methods, various algorithms, lot of computational work is going on in this area, and specifically the IIT Hyderabad is who is also the national coordinator of the GYAN course. They are extensively working in this GNA quadruplex structures, computational structures. So computational algorithm, various algorithms, they can be predicted by the G4 formation based on DNA and RNA sequence, in vitro characterization, UV melting experiment, CD spectroscopy, gel electrophoresis, fluorescence, etc. So many techniques are there for in vitro characterization, which will let us know whether it is a parallel, anti-parallel, double strand, or it is a quadruplex kind of a structure. So they will give us an idea what kind of structural features are there. And finally, atom to atom or molecule to molecule coordinates, whether it is in the form of nucleic acid bases, sugars, backbone atoms, their XYZ coordinates, they are being confirmed by NMR and X-ray crystallography. So biological significance of G quadruplexes, or you can say the G tetrads, are they are they are confirmed to be found in the in abundance in telomeric junctions, which are the protective caps at the end of the chromosomes. They regulate the expression by influencing the transcription and replication. G4 interfere with the DNA repair pathways, potentially causing a DNA damage. It also it has G4 has implicated in cancer development and progression, making them a potential therapeutic target, and where my research work or my research lab and my students, PhD as well as postdoc students, they are working. So these are the references or the research papers, review articles, and the books from where I have compiled this lecture for you. So thank you very much.
Thank you, ma'am. Thank you for such an informative talk. And I wish you all had an inspiring and insightful lecture. Now, next, we will.
Any questions? We'll be having tutorials on
Any
this. We
questions,
will be,
anyone?
yeah.
It's not a question,
Yes, ma'am.
just a comment that in on 15th I'll be giving a lecture about the applications, how we can determine these structures. So exactly whatever Dr. Pamita has said, I will show you how by using circular dichroism spectroscopy and NMR, nuclear magnetic resonance, we will be able to tell what are the structures and how ligands bind to it and how useful is this for our medicinal biology. Thank you very much, Dr. Pamita, for a nice lecture and introduction. And I will be speaking, just ahead of it, whatever she has said in the last slide. That is application by circular CD spectroscopy, thermal melting studies of G quadruplex DNA, and NMR based structures. Thank you once again.
I'm honored, ma'am. I'm speaking because you were my mentor, you were my guide. So thank you so much, because I'm here because of people like you who mentored us. I think I started my postdoc work from DNA duplex NMR and molecular dynamic studies. So thank you so much, ma'am. I would like you to comment if you find any flaw in my lecture or in my presentation. So I would like you to let me know that also.
Thank
So all the participants are requested to join at sharp 2 p.m. We have a lecture from the foreign faculty from University of Strasbourg, France.
Any
Okay, so
questions? No
kindly get
questions?
back all of you at sharp 2 p.m. Okay, thank you everyone.